guys, it's so nice to see you after a really long while. So in this video, we are going to see a short topic which is called as a superbug. More people may or may not know about this topic. Let us go into the topic. Before knowing about what superbug really is, we need to know something about the term bioremediation. So from the term, the remediation is giving remedy to some kind of problem, and bio in the sense we are using biological thing. So here we are cleaning up the pollution with the help of a microbe. This is called as the bioremediation. On speaking of the superbug, it is a constructed bacterium which was artificially made. In 1979, a scientist called Anand Chandrabharti found the oil spill a greater problem. So he wanted to find some remedy towards those problems, and he founded a bacteria which contained 4 different strains that can clean up the oil spills. Commonly, a petroleum contain organic and as well as inorganic compounds like naphthalene, toluene, xylene, and more aromatic hydrocarbon, which had become a greater problem to birds, animals, and so on. So this has to be cleared. So he found a plasmid. Let us see how he constructed the plasmid below. So the construction of superbug includes the identification of parent strain. He has to identify different strains. Then he had to transfer the plasmid. He has to conjugatively transfer by means of mating or some other process. He has to transfer the plasmids. Then he has to select the plasmid. Then culturing it in an enormous number. So let us see each steps one by one. Identification of a parent strain. First of all, we have to identify different things. He found 4 different things, like 4 different organic matters, a big problem. Those things are camphor, octane, xylene, and naphthalene. These things has to be removed. So his constructed bacterium contains 4 types of plasmid, namely camp plasmid, which can degrade the camphor compound, oct plasmid, which can degrade octane, hexane, and decane compound, xyl plasmid, which can degrade xylene and toluene, and NaH plasmid, which can degrade the naphthalene compounds. Then the transfer. Here he chooses 2 strains of bacterium, namely A and B. The A strain consists of camp plasmid and B strain consists of the OCT or oct plasmid. He made to mate them both, and as a result we get a plasmid called cam-oct plasmid, which contain a cam strain as well as an oct strain, as you can see both colors here. In the same way, he took a strain C and strain D, where the strain C contains xyl plasmid and strain D contain NaH plasmid, and both of them are being mated, and we get a strain called as F, which contain C as well as D, xyl plasmid and NaH plasmid. When these 2 things are made to mate each other, we will get what we needed. Which is nothing but our superbug, which contain all the 4 things like cam, oct, xyl, and NaC. All the 4 things in a same bacterium. So this is how our superbug is being made. So here nothing but we are mating A strain and B strain, and we get E. Similarly, we are mating C and D, we get F. When E and F is being mated, we get G. On understanding the picture, you may find it even more clear. So now we get a plasmid called G. Our superbug is being produced. So the next step is the selection of superbug. Selection in the sense, the superbug which we get is to be selected for mass culture. So we are culturing it. Now we get our thing, and now we are adding a new medium to grow, which means we are subculturing it. We are growing it in another medium so that the desired amount of superbug can be produced. On seeing into the application of this thing, Anand Chandrabharti got patent to use the superbug. In 1990, the American government allowed him to use the superbug to clean the oil spill on the water of Texas state. First of all, we have to subculture it. Then we have to mix them with a paddy straw. Then we have to keep them in sunlight to dry, or somehow we have to dry it. Then we have to spread on the sticks which contain oil, and now we have to put it on the place where it has to be grown, and finally it will break the oil into non-polluting materials. As a result, the oil waste become harmless. So these are the things about superbug, and finally it does not pollute, or rather it break the hydrocarbons into soluble forms, and it is so helpful for us. So that's all about this superbug. Anyway, thanks for watching, and thank you all.